Second, I thought you meant me. I'm like, already heard that today.
What?
Jaws are upside down. Okay, zoom out. The shrimpy guy. I'm excited to get to the edge here. It may not happen on the spot.
I have
the edge
bad
should be
news
fun.
for you.
Yeah,
we are
the
not
edge is going to be really cool.
Circumnavigate the edge.
Yeah,
This is kind of,
it's a little sparse.
yeah.
That depends a lot on the science team, because sometimes they actually want to see that there's actually nothing up there.
Prove that
Yeah,
there's nothing.
nothing is a valid answer, for
Yeah.
sure. That is data. And it's not really nothing. Sarah was saying it's something.
Yeah, there's a lot in that sand. That crab's not eating nothing.
No,
What do you
that's
think
true.
he's eating?
Probably like little critters that are living in the sand. You can look and see all the different colors we're seeing in the sand. Probably lots of different
Video, can you give me a zoom?
worms and
Just
stuff.
like zoom. Just go for broke. Here, I
little
will.
snails.
I'll bring it a little closer, so there we go. See if you can get a focus. There, nice.
Oh, there was something.
Going too fast.
Bridge
1
nav,
second.
step again, please.
Try it. Can you go a little wider?
Yep.
Try and steady us out a little. That's nice, right there.
There's
Oh,
something.
there's a brittle star.
Oh, whoa.
Nice.
Are brutal stars crab food?
Crab food. I don't know. Brittle stars are pretty decent predators themselves, aren't they?
Yeah, and they can
Yeah.
move quickly,
who
so
would
I'd be
eat who
surprised if
In that fight.
that crab was eating a brittle star.
In a face
but
off between a brittle star and a crab.
There's
Yeah.
a snail.
So brittle stars can like essentially pull a leg off
Whoa.
with a predator and then regenerate it.
Right.
So that's probably,
Can
I'm
you
guessing
try and
that's what the
refocus?
outcome would be.
Yeah, perfect.
I think when you see areas like this, though, you also really appreciate why things like whale falls create such a feast in
Yeah,
the deep
right.
sea.
Totally.
Difficult down
Okay,
here.
go wide. Catch up a little.
What's that up there?
yeah, what is that?
Ooh, what is
Big
it?
fish.
Whoa.
Hello.
Yeah, if we could get a nice zoom on that guy.
What is it? It's cool.
Yeah. It looks like an eel or something. What?
A maritime danger noodle.
Go for zoom video.
Whoa.
Oh, it's one
Wow.
of those, yeah, what are they called?
Wow.
Oh,
That's
nice.
awesome. He looks grumpy.
Yeah.
Looks like he's like made of stone.
I think we're very critical about all the animals we see.
I know. I always
We're
like
like,
assign
"Oh,
a
he looks
mood to it so
grumpy."
quickly.
Yeah, this giant screaming idiot thing in front of me.
Oh, the Argus shot is pretty cool.
Yes.
Fish staring
Oh
down
yeah.
Hercules.
Little stare down.
Do you want me to pull out a little bit?
Yeah. Come out a little. Yeah, nice. Good shot.
Wow, that looks cool. Oop.
Okay, go wide. He got wise to us. We are foe, not friend.
You scared him.
No, I think we just won the stare down.
This is a little unfair. We're so much bigger. It's scary.
What do you suppose did this crater?
Something
Oh,
making
yeah.
a nest.
Investigate.
Oh, that's
Could have been a
weird.
fish.
Video, can you do a
But I
zoom
don't
for
know.
me?
Yep.
Or it could have been like a burrowing shrimp or something like that, and it kicked up the
Yeah.
sand as it was burrowing.
There is a shrimp in there. Do
That's a-
things make like little sand traps like this so that other things sort of fall in?
On land, I'm trying to think of any marine examples of that. Can't think of any.
You can go wide.
Yeah, on
'Cause, like,
land you
you
have
can-
the like antlion traps that
Okay.
ants will fall in, and they can't. It's too. Guess it's too steep for them to climb out. I don't know.
Because they definitely gathered
And
like
take
a
your
whole
bubble
bunch
can.
of stuff. Yeah, go for it. Antlion, yeah.
Okay.
Yeah, it, like digs a little
Yeah, I've seen it. Like,
cone-shaped hole.
no, no, I've seen it. Like,
and it
I've
lifts
seen
at
it.
the bottom.
There's several places nearby where I live that there are these kind of ants, and you can see they're like this big, probably only
Oh,
the
antlion
craters.
is?
Probably terrifying if you're an ant.
Yeah. But I didn't know the translation was so straight, because for me, it's literally an ant lion as well. Like the same ant and lion translation
Go for
is.
zoom video.
Breach nav. Step again, please. Thank you.
Okay, go wide. Oh, hey.
I just started, but good.
So we're starting to
Hand it over to you.
transition
I was
into
just
our
taking the
watch
watch.
change right now, so you may hear a little bit of silence as we catch up the new Nautilus folk entering into the room. But thanks for joining this watch. We had a great time, and we'll continue to dive for a few more hours, so keep watching.
Yeah, it's pretty steep on the south part. Yeah, just so. Because I don't know what contour will be our target. But right here, it's like super. I think so, yeah. Rock targets, okay.
Maybe we should check with Alexis first. I think that
Sorry,
she's
go ahead.
brief.
I can't hear you.
So we're moving due south until we hit a feature that we want to follow?
Ben,
Yes.
it's kind of hard to hear
Really,
you.
we're just looking for hard substrate. So let's
see.
just kind of, we can look around here and then essentially continue along this contour if we think it is going to continue with rocks.
Okay. I imagine that on that steep slope it might start to get rocky, but it's
Right.
pretty steep.
I know. I don't know how much we want to actually like drop back.
Okay. Well, I'm going to keep up the ship moves if that's
Yeah.
cool. Okay.
Sounds good. Yeah, so we found some hard substrate again, so that's a good sign.
So Alexis, you're looking for hard substrate but not vertical cliff surface.
I mean, vertical cliffs are fine. I just don't want you guys to have to drop down too much if that's an issue. I can't hear you, Hark.
Wasn't talking to anybody.
Oh, sorry.
Yep. I was just commenting to myself. You know. People would say things, I would comment.
Really funny jokes, I bet.
It was hilarious.
It
You should have been there. Wish you could have heard me. We're fine doing vertical cliff surfaces.
Oh, cool. Okay.
It seems like we want to kind of stay towards the top, though, right?
Yeah.
Like on this fringe. Yeah, it looks like there's another 20-meter drop. That's pretty gradual.
Big fish. Thought it was a rock.
Oh, yeah. You see a shadow.
Hey.
You see a fish
Oh,
in
wow.
here? Oh.
He is a
like
big
a
fish.
Cuskin or?
Is it a rat tail?
Is
Everything's
it
a
a
rat
tree?
tail.
Everything's
if you
a
want
rat
it to
tail
be.
to me.
Yeah.
That is a big fish,
It's a good
though.
go-to. Oh.
Oh, tether extent.
Here, I'll drop down to something
Video,
lower.
let's get the fish.
He's a chunky.
I believe the technical term there is chunk.
You can see why it looks like a rock. It looks like stony scales in it.
Yeah.
How old is that fish?
Probably old.
How old do these kind of fish live?
I don't know.
Do we know?
Sorry.
Older than trees.
Oh, she's already back on
to
it.
the trees. That didn't take long.
First 3 minutes.
That wasn't even 3 minutes, guys. That was pretty,
Yeah.
pretty immediate.
Oh. Later.
Later.
All right, full wide video.
Little Umbellula sea pen there.
It's like 40, 50 centimeters.
Yeah.
Big
Big
fish.
fish. Must do well out here.
Come on, predation event.
For science, of course.
Well,
Oh,
there's,
yeah.
there's truly not a lot of predation events recorded. You know, like, you know that they have to be happening.
Right.
was on a watch, and the entire dive ended up being called the predation dive. There were like 6 or 7 predation events in one dive.
Oh, man.
Deep? Yeah. Because I think the overlap between predator and prey is just rarer to these environments.
This guy's fighting for some camera time, too.
He's like, what do I have to do to get you guys to pay attention to me?
Yeah, I thought about it, but we would need a little bit more tether to chase him properly.
Yeah. Sorry.
Come on, Argus.
How are our numbers, Jess?
Looking good. Everything's stable.
That trail looks bioturbation-y. Yeah.
One of my 2 favorite words.
Bioturbation.
Bioturbation and coddle peduncle.
Coddle peduncle.
Oh, man.
What was that one from again? That's from a sea pen, right? Coddle peduncle?
It's a fish.
Coddle peduncle, I think, is specifically fish, but a peduncle is found on a sea pen. Is that correct?
Mm-hmm.
The bit that you nudged would be the peduncle.
The coddle peduncles used on fish. It's curious.
I mean, if the peduncle for the sea pen it's kind of an anchoring device, does it have a similar function on a fish? Do we know?
It doesn't work as an anchor.
No?
What is it?
It's like the fleshy part of the fish right where the tail fin attaches.
Oh.
It's like where the notochord ends or something.
Oh.
Something like that.
So it's just like an anatomical marker.
Yeah. Well, it's a point of flexion, I think.
Okay.
For the movement of that fin.
Oh, these guys are pretty.
Yeah, sea lilies. We've seen a bunch of them on this dive, and different kinds.
Should we do another round of intros now that we've got everyone kind of swapped around?
Yeah. Front row?
You got it. This is Brandon Rodriguez, the Science Communication Fellow.
And I'm Marley Parker, the video engineer.
I'm Jess Sandoval, sitting Argus.
Greg Diffendale, sitting Herc.
Ben Gracian, sitting in the nav seat.
I'm Megan Lubekin. I'm on data.
And this is Holly Richards in the science enthusiast seat.
And Alexis Weinig in the watch lead seat.
So we're hanging out on top of the seamount right now, and
wait. We have one more person.
I know, but he shook his head.
Oh, he didn't want to say anything.
Oh.
Hi, I'm Ben Craig. I'm a data engineer.
Yeah.
Yeah.
You're important too.
Ben is very important.
He keeps the internet pumping for us.
And all the other things.
And everything pumping.
He's a bit wrangler.
He's a bit wrangler, exactly. Makes the best winch GUIs in all the land.
Thank you. I'm going to ask Justin if I can change my job title to Bit Wrangler.
Yeah.
Yes.
I mean, like, data engineer sounds good, but, like, bit wrangler is, like, now I'm curious.
Oh, let's take a look at this fish on the left.
Yep.
Hi.
Ooh, frogfish, huh?
Ooh, looks loby, yeah.
Yeah, loby.
Right now he's like, I hope they don't see me.
I'm camouflaged, right?
I'd like to see this fish make an expression that wasn't that.
Is it a frogfish?
Yeah.
So.
Place.
Tell me when, Greg.
In the genus Chonyx.
Is it smiling or frowning?
All the way down.
I think they're always frowning, right?
Or always
we
smiling.
saw a
You saw a
fish,
smiley
a
one?
fish that looked like a frogfish. on the previous cruise, and it was crazy smiling.
Oh.
Really?
Yeah, but I guess it wasn't a frogfish then.
I mean, I don't know.
They can
I
only
always
drown.
confuse the frogfish and the batfish.
Or what?
The antennareids,
The
and the
batfish, you said?
oxyphalids, yeah.
He's got a very grumpy cat vibe going on.
Oh my gosh.
Vibration. Sorry, dude.
Hey,
But
buddy.
Okay. He's off.
At least we know it's capable of movement.
It's capable of movement. I don't think he would make it down here if he wasn't capable of
there it
movement.
is.
Oh.
Yeah, he's just swimming.
Hi.
Doing your thing.
If you didn't think I was a predator before.
Might run out of leash pretty soon.
Even though it says it on your arm.
We're going on a guided tour.
You can see its little lure. See the?
Oh, yeah.
So they're technically a type of anglerfish.
Oh, really?
Yeah.
Oh my gosh.
So that's what's popping up the
Yeah,
little
right on the
thing
front.
right on the top of the head.
Yeah.
Does it have a
All right,
little light
Marley,
bulb?
let's go in.
I don't actually know. I just know that they. I mean, I think so. It has that they're there generally to attract prey.
Mm-hmm.
Probably feeds while sitting on the
Such a wild
Yeah.
texture.
Yeah.
So it would just sit there and like kind of project that.
Out of tether.
You can see its fins where earlier it was like perching on them. He's kind of just gotten tucked back.
Okay.
Really see the caudal peduncle on it, really.
It's got a little shrimp.
So anglerfish as a category are the ones that typically have some sort of lure that they're hanging out front to bring in prey.
Right.
No, some of the ones that I've seen are bioluminescence. Are they always bioluminescence?
I don't
the
actually
lure?
know. I haven't
Alright,
studied
that's
fish
all we
enough.
Okay.
Coming out.
That's a good little swim shot.
Yeah.
So how do you make a frogfish
Thanks, video.
smile?
I don't know.
Is
Is
this a
that a joke?
No, I'm curious now. Someone
I don't
said
know.
they saw it smile, and I'm just.
Wait, no, okay. It wasn't. It must not have been a frogfish if it was smiling.
I mean, it could have been. It just had its mouth was oriented
Yeah.
differently.
Jess, do you remember? It was on our watch last cruise. That crazy smiling fish.
I wish I did now.
I have a picture of it. I will show you later.
Please do.
Found some rocks.
Nice.
Mm-hmm.
Follow the fish to the rocks. Oh, there he is, perched over there too. You can see him using his fins. Right past the lasers.
Yep.
But
Yep,
so
yep.
we have to get back in front of everything.
Yeah, you guys.
That shrimp is still looking for some camera time.
All right, give the shrimp some camera time.
Come here, shrimpy.
And now he's shy.
Our community is saying it's a goosefish, not a frogfish. Is that correct?
Oh.
Do we know?
We're
Sounds
looking.
reasonable.
I think that sometimes.
Okay, full wide.
I'm not positive on the common names for them. They might be interchangeable.
And then, yep, thank you.
Google image which one's smiling. That's how you know.
Yeah.
All those fish are these like lophiformy, lobate fishes. Like the scorpionid, like the scorpion fish are in that category. I mean, it's a big group, but
Notes.
they have
Ideas.
similar
It definitely
morphologies.
looks
They're
like a
definitely
goosefish to me.
Yeah.
But both frogfish and goosefish are anglerfish.
Yes.
Deep sea goosefish.
But thank you, people at home. We can always use help with fish IDs.
Yes.
We do not have any fish biologists on board.
All right, bat signal's going strong.
So we're cruising around the top of this unnamed seamount right now. Are we looking to kind of get to the edge? Where are we heading from here?
Yeah, so on the last watch they'd come up and the top of it is kind of a flat space, so it's just been sedimented, so it's like this, really sandy. Well, this is an
I've
interesting
enjoyed my
habitat.
day
It's not
completely.
exactly what we're looking for. We're looking for more of those hard rocks where we're likely to find corals and sponges. So we were working our way to the south end of the top of this mound, where we'll hopefully find kind of the ridge, and we'll have a little bit more of that steepness that we saw as we were coming up.
Yeah.
Like these lovely rocks.
Ooh.
Lots of stocked crinoids. We've collected one pretty large one that had 5 arms. But we've seen a number of different types.
A viewer from the UK is wondering if this is a good spot to find cephalopods before we move back down.
That is a wonderful question.
Yeah.
If we find one, though,
It's
stay
definitely a good
tuned.
spot. Yeah.
That's true. If we find one, it's probably a good spot.
It's a good indicator.
Yeah, they did. They saw one on the last watch.
Yeah, I think the Dumbo octopus count is 3 for this dive.
Oh, yeah.
Is it really?
Yeah, just not our watch.
Oh.
Wow, wow.
Yeah, well, let's hang out and look for them then.
So many of these crinoids.
These crinoids are so cool.
They're all facing the same direction, too.
Yeah.
Which is interesting to think about. Means the current is probably flowing towards us. And we are in kind of the last portion of our dive today. We're going to be recovering in probably a little under 3 hours. So we've been down for 20-ish hours?
Yeah, actually, because we started it yesterday at 8 p.m.
Right.
Yeah.
Okay.
So 24 hours since dropping in the water, right?
Yeah.
We'll recover, yeah.
Right. Will be our total dive.
Yeah.
Fun.
And then we'll head tonight. We'll steam to we'll steam to Baker Island. We'll do some mapping when we get there to fill in some gaps and collect a bit more of the bathymetric data there, and then we'll hopefully be launching at around 0800 local tomorrow for a dive, a 20-hour dive at Baker.
And so Baker Island is one of the national wildlife refuges that are here inside the Pacific Remote Islands Marine National Monument, which is some of the areas that we're exploring on this set of dives. And the Pacific Remote Islands Marine National Monument is one of the largest protected marine areas on the planet, with over 400,000 square miles protected, and 7 national wildlife refuges within it, including Baker Island. So pretty excited to actually go up that seamount, because I think we're going to be getting fairly close to the surface on that one, hopefully.
Yeah, our target track is around 2,000 to 400 meters for tomorrow. So covering quite a bit more vertical distance than what we were doing today.
Still pretty deep.
Yeah.
Yeah. It feels close.
Is it, we're off the coast of Canada and Mike was piloting and we got up to 15 or 20 meters, something like that.
Oh my gosh.
He was within the wash. You
Wow.
know, like,
wow.
yeah, it was pretty neat.
The sides of the seamount for Baker Island are really steep, though. There's not a lot of terracing near the top or like nearshore coral reef environment. So getting up that high, even though we should be close enough to the island to actually see the seabird colony out there, which I'm pretty stoked about.
Like from the ship?
Yeah, from the ship. So we should be able to like capture some good video of the seabird colony that's there right now, and get some of that data. So the U.S. Fish and Wildlife Service manages Baker Island National Wildlife Refuge.
There's a shrimp out there.
But it's so remote that we can really rarely get out there to do any sort of like rapid biological assessments. So even just from the shore, being able to capture some video of the seabird colony will be great data for our biologists.
Oh,
now he's gone.
The escape response.
Full wide.
How often do people from Fish and Wildlife go to these islands to do some like biological surveying and things like that?
It's almost never more than every couple of years, and just doing like a rapid assessment. And it's normally if we can catch a ride on another boat that's going by.
Right.
So what that means, though, still, is that it's really hard to approach these islands because they don't have much of a fringing reef at all. They're really small, and they're in a big like current area that it's really hard to approach them. So sometimes we'll get out there, and if the conditions aren't right, you know, we just hitched a ride 5 days on a boat for nothing, and we have to wait till the next time we can get out. Yeah.
So the seabird colony is a single species? Or?
No, there's a number of species that are out there. So some of the really big species on Baker and Howland are lesser frigatebirds, masked boobies,
You and my
blue naughties.
Fish on the right. We can
Oh,
zoom
we
on.
can talk about
while
fish, too.
No, we can totally.
Fish versus birds right
we
now.
can
No.
zoom
Seabirds are marine
and
mammals.
bird talk at the same time.
Seabirds are marine mammals.
All right, video.
Well, they're not, but
I was
you
like,
know,
what?
They're not.
We're not mammals.
They're not mammals.
I was like,
I mean, they're a marine species.
Yes,
I find
they're
that
marine dependent.
Here's a little fish.
Fish is like a fencer. It's got a sword.
Yeah, I got a
Yeah,
little
he's got
spine
Yeah,
out the
it's
front.
got a
Dorsal
couple
fin.
interesting.
Spine on it.
Big eyes.
Looks like a
Looks like a
catfish
wizard
light.
fish.
Oh.
Wow. Wait, look, you can see it moving its little thing.
That's the scientific name.
Biology is a big field, right? We can't know it all.
True. That's what I tell myself.
Oh, I like it.
This guy's really
Oh,
cute.
yeah, it's
Super
nice.
cool.
It looks young, doesn't it? Yeah, maybe not.
Look at those sensory organs around the mouth, right?
Yeah.
It's really cool.
This is a good shot, too, for people that do study fish to be able to ID it later on.
I love fish that have these dorsal rays, like the fans. So.
Mm.
Wonder what he's looking for.
That's great, whenever you guys are good.
Sure. Video full wide.
Coming out.
You know, that's the only rock pen that we saw on that watch.
I know. Oh, yeah. That's right, yeah. That's a good point.
It's a good thing you got it.
Really wanted to take a look at its suction disc when it gets on board.
I was surprised they actually let go so easily. Sometimes they don't let go. Like they're just, no.
Yeah, they refuse.
He was ready.
He was ready. It's like, take me to the science.
I'm a believer.
New life, new me.
Because Jess, you do work on like biomimicry inspired adhesive stuff, right?
Yeah.
Stuff.
Yeah, I do things with, biology, biomimicry, biomaterials. So suction discs are of great interest to me.
Super cool.
Adhesive mechanisms and attachment devices of creatures are always things I enjoy studying. Doing research
Especially
on.
things that can release, right?
Yeah, things that are non-permanently binding, or you can apply those to anything that requires attachment but not irreversible attachment. So manipulation, locomotion all require you to detach from surfaces in addition to attach to surfaces. So so when you guys bring it aboard, you know, let's take a look at it real quick.
Yeah.
Out of curiosity and fascination.
We'll try and keep it in water, too, because it might look quite different when it's out of water.
Dehydrate, yeah.
Yeah.
Have you had an instance of kind of discovery here that's helped your research? Do the two overlap?
Yeah, sometimes I look at samples under the scanning electron microscope back on the campus that I work on. And a lot of actually the work kind of started with, I asking myself how things delicately grasp, because we don't delicately grasp out here. Well, we do, but it takes a lot more effort and skill with the manipulators. So the interest in delicate grasp and eventually this biomimetic attachment actually came from me sitting here and realizing that it's actually very difficult to manipulate things with a delicate
Video,
grasp.
let's push in.
So very influential, if you'd say.
And turn around.
Right?
we
Wow,
go.
Greg.
Yeah, he's still in training.
I can't tell if
Is
those
he
are
swimming
fat
vertically, or is that
pectoral
just the
things.
I think
angle?
it's just. I don't think he's. Yeah, I think
He's
it's the
he's
angle.
slightly vertical.
Is he? Yeah,
But he's not
But yeah,
vertical.
Okay.
he's just kind of cruising. Looks like maybe.
It's a rat tail.
Right.
but-
Yeah.
And I think it's an Azumia, is what those are called.
All right, full line.
Rocks.
So Jess mentioned taking samples back. What kind of facilities do we have here, and why is it important to kind of get started on the science versus haul things back home?
Well, it kind of depends, like what we're working with and how much we get started with the science. On a cruise like this, we're mostly like preserving samples for the primary analyses to be done back on shore. We do process eDNA, so we do filter water, which is kind of an environmental DNA sample near a coral. So that happens on the ship. And sometimes we do a bit more in-depth processing depending on the cruise, or occasionally we'll have an additional lab on the ship.
Yeah.
But with most of the samples we'll be collecting for this cruise, we'll be looking at rocks and corals and other biological organisms, and pretty much documenting them and preserving them, and getting ready to do more diving so that we can make the most of our time while we're out here in this remote area and save some of the laboratory science for the shore.
Yeah, we have to remember that we spend however many hours on the seafloor, and then especially our data managers, and then myself will be in the lab for a couple hours after the dive as well to process the samples. What's that on the right?
Fish.
Oh, it is a fish.
Yeah.
We can take a quick zoom.
Greg, am I
All
good to
right,
zoom?
Marley, go.
Ooh, it's an old grandpa.
There's definitely a greater abundance of fish on the summit than
Yeah.
on the slope. Oh man.
That one has some battle scars. So I was like,
Yeah.
That's all we're gonna get.
This looks like it has a couple of parasites too on the fins.
He's seen some things.
Excuse me.
So we're pretty much, this is the edge.
Yeah.
I see. Okay.
Oh, wow. Into the abyss.
Might be another like 20 meters.
Yeah, we'll probably want to position Argus, bend Argus a little bit deeper, and then
Yeah. Yeah, if we're
we'll
gonna
kind
do
of
the
strike it along the side.
That one, like crinoid stalk
So
that
I'm
doesn't
gonna
have
do
a crinoid
Yeah.
anymore.
Aw.
Stock.
Sorry, dude.
Yeah, 20 or 40, either way.
Out of stock.
Let's go to a different depth.
No, it's got a lot of stock.
I think you would say it is in fact stocked.
It is in stock.
It's in stock.
It's in stock. If we can get a close-up on one of the yellow fans, just because
Okay.
they're kind of all over, but we haven't really taken a close look at one.
I was looking at the rock C-pen.
Oh, yeah.
Oh, yeah.
That's like a different.
We're going to do an up and spin.
Not sure how much tether you'll have. I can come down a bit more.
Yeah, I can come up a little bit towards you. All right, Marley. And I'm stable, so you can go all the way in and get your focus, and then.
Cool. If we can just put the lasers on, or do we not see them when we're zoomed in?
Yeah, we don't
okay,
see
cool.
them when we're fully zoomed in.
That's fine, as long as they were on earlier.
Yep. All right, let's back out and frame it. Lasers.
Great. Gives us a good representation of the branching pattern, and that close-up on the polyps is helpful. We see a lot of these kind of around this area, but it helps to have a nice close-up for better identification for people that use this for kind of species distributions.
Looking at what we've sampled so far, is there anything that we're missing, anything we're still hoping to see before we end the dive?
We do need to fire one more Niskin, at least one more Niskin, and have it be where we have an area where we don't see a lot of corals on rocks as a control for the eDNA.
Yeah,
but
we can do that on the way up a little bit too.
Oh, okay. It doesn't need to be like on the seafloor?
I think it's better if it can be, but considering that when they were on the summit, they didn't think it was appropriate, I don't think we'll find somewhere around this area.
Science, can you guys talk a little bit more about eDNA and how and why that's important? It's a relatively new thing in oceanography, right?
Yeah, so eDNA stands for environmental DNA, and essentially what that means is just detecting DNA particles that are within any substrate of the environment. So you can get eDNA from mud, like down on the seafloor. You can get eDNA from water. You can get eDNA from soil on land. And what the idea behind it is really to be able to detect what animals are around, or organisms. It could be plants when you're on land, or have been around recently. So it's just filtering out the DNA that's in the water is what we're doing. And then it can be sequenced, so the DNA can be identified back on shore to see if we're picking up the same coral tracers that we're seeing the live corals when we're down here.
Oh, sorry.
And so the idea behind it is to be a more non-invasive way to sample, so that you wouldn't have to come and like take actual snips of corals every time, but you could just take eDNA samples to get a representation of what that community might be like.
So right now, are we still in the stages of sort of verifying that?
Yeah, and like really
We
to
can always just travel
I can drive
to
upslope a little bit.
and we can face him.
be able to reference your sample, you need to have a database, and so we're still kind of building that database of barcodes that you can, so you can identify what the DNA is.
Little feet.
Yeah.
Does the presence of all the particulate, all the sea snow that we're seeing, does that confound the eDNA results?
I don't actually, I've never run eDNA myself, so I don't know how much that can impact.
I don't think it's a huge deal. We're seeing
yeah,
rain
the
snow
super
kind
tiny
of
opioid
all
in
over the
the corner.
place.
Yeah.
I haven't.
It's
I've
like-
done the front end of eDNA a lot. So
Yeah.
taking water
Like right by
from
the sandy section.
from the deep sea and filtering it and preserving the filters to be analyzed by Meredith
Oh,
Everett.
it's got this giant thing coming after it.
Who's the scientist kind of building this database.
All right, video, let's
Yeah.
come full wide.
But it's a good question.
So, Alexis, are you happy with this contour here? If so, we'll reorient Herc and Argus and then go along this contour?
Yeah, I think so. If you guys feel that that's good too.
Yep.
Yeah.
Cool.
So,
I think
Jess, you want to follow me around?
Yeah. Let's see if I
I think eDNA is pretty cool.
Port, so
Also because
that
it's
gave
not
us
just
more of a tether wrap.
non-invasive, but it's also gives you the ability to glimpse into what might have been here without the vehicles, because we do bring lights and noise when we come marching through
I might
here.
go to
So
starboard just to take it out.
it's kind of a
I think I should take it out.
secret way of checking out who's been around.
Yeah, and it gives you like you can take water samples without always having an ROV at the bottom. So
exactly.
if you're doing CTD casts or something, you can filter for eDNA that way too. So it can be actually a lot less expensive to come and do it.
And because like organisms are
working.
constantly sloughing
There's
off
some more.
DNA, so it's all of the organisms that are theoretically in that area. Do we have a sense of like how big is the
They don't
area
always
that
work.
we're sampling when we're collecting
Brains?
some eDNA
Yeah.
from
Alright.
somewhere? Do we know where
But
all it could
I
be
think
from?
that it's so related to the currents and how things are moving around, right? So
Yeah.
probably really depends on where you're sampling.
And that's one thing that
How long is the eDNA good for? Like,
yeah, so that's what I was going to talk about, is we actually have a collaborative project with some people at Lehigh University, so Santiago Herrera and Jill
video, let's push
McDermott,
in. Baby sea star.
to run some laboratory experiments to test eDNA stability. So we have tanks in our lab at Temple that have live corals in them, so we know that there's eDNA in there from those corals. We've developed biomarkers, or we've developed primers for
That's all
that
those, yeah.
specific coral, and then we put the water under different treatments, so different temperatures, different pHs, and different oxygen levels, and see if the DNA
Yeah, I'd probably go
degrades at
east.
different rates.
Confirm.
Um,
Baby shrimp.
but we haven't run all the experiments yet because I'm here.
Hey Alexis,
Yeah.
are we good following
All right,
this
Marley, let's
contour
go
to
full
the
wide.
east? Kind of
Yes.
Get depart from where we've been?
Yes.
Okay, great. All right, I'm gonna ship move back.
So is it fair to say right now then with the eDNA that we're still figuring out like what are going to be the broader implications of this data, or are we still figuring out how it can be used in the future and building that database and building sort of a fuller understanding?
Yeah, I think so. And again, like this isn't really stuff I've worked personally on, but I think in the last like 5 years, really, it's developed a lot, and in the next 5 years, it'll develop a whole lot more.
Yeah. That's cool.
I think it was like 3 years ago we started doing it pretty regularly out here, and one thing that we were doing a lot per Meredith Everett's advice was
Video
collecting
shrimp.
a water sample directly next to a coral and a little bit further, and trying to see like kind of the signature of that coral in the water at different places. So I think that's a pretty useful way.
I would probably just go due east. I think that's
Finally
fine.
got his 15 minutes.
You know, we may go deeper.
It's that
we
same
may-
shrimp.
He's
Almost
been following us around.
It's a good
All right,
dance
full wide,
move.
Marley. Thanks.
We talked our last shift about having, some sort of AUVs or remote kind of operations.
Yeah.
I wonder if
right.
eDNA would be one of the things that you could kind of just keep monitoring down there, right? There is flow, and you are kind of sampling a large area. Why not just have one device left behind? Is that something that's possible? Do we ever leave something for readouts at the ocean floor?
Well, we don't have any way of like getting a readout essentially underwater. So what we do is we filter the water, and then that filter goes back to shore to get analyzed. So we're not to the point of having a sequencer on the seafloor telling us what's in that sample, even though that would be amazing.
That would be so cool.
But in terms of that, you do have the mass spec and laser spec, which we've brought down on the vehicle. So it's not the eDNA, but as far as real-time analysis. Yeah.
You brought the mass spec down like on the ROV?
Yep.
Oh, wow, really?
Yep. Yeah, we've done a mass spec, a laser spec.
What did you use it for? Like what samples were you looking at?
Stuff.
Specking. In specking.
Chemical species in the water, I guess.
Oftentimes it's used at flow sites where there's some kind of venting. I think we also used it in brine pool stuff.
What do you mean when you say a flow site?
Hydrothermal.
There are sites, all over the ocean floor that emit either there's low temperature flow or high temperature flow. High temperature flow is usually bursting out and can be, up to 400 degrees C is I think close to the highest that we've measured.
You like thermal vents?
Yep.
Okay.
And then the low flow is kind of more of a seep. You can maybe see it visually. Sometimes it's very hard to see visually. But it's the chemical differences in the water that allows you to see it, because you'll see just kind of like this little bit of shimmer.
Yeah.
And so we'll try and find that vent and stick the mass spec sampler in it.
Well, that's pretty cool.
Who developed, which one have you used with?
We've used, why can't I see his face.
What was the question?
His face.
Who developed the mass spec for depth?
Scott.
Scott spec.
Scott spec.
Might be a good question for our
Stephen
viewers.
J. Spectroscopy.
No, if he's listening, he's just probably like mad at me for forgetting his
Greg,
name.
how could you forget me?
I know.
Do you know what institution he's at?
Yeah, Hui.
Is it Scott Wankel?
Yes.
Yeah.
Wankel.
Wankel.
We named a hydrothermal vent after him, I think.
That's cool.
We called it the Bull Wankel.
That's awesome.
That's funny.
It looked like a moose head. It was like this one chimney that then went up and like flared out to a bunch of little chimneys.
That's
Oh,
cool.
wow, that's amazing.
You're saying it was very rocky at the Bullwonkel?
Oh no.
Oh, no.
On point.
On point.
Ben liked that one.
Oh, yeah.
Speaking of names, though, this is an unnamed seamount. So do we get to name it? Who gets to name it?
It's actually it's a process to name a seamount. So
Really?
yeah, you have to have a lot of data about that seamount.
Well, and this seamount is outside of U.S. territorial waters.
It's technically in our EEZ.
Oh, it is in the EEZ?
Yeah.
Okay.
It's just outside the
It's
Baker
just
Howland.
Right. It's outside
Yeah.
Prim.
So it's inside the exclusive economic zone of the
Right.
U.S., but it's
Yeah.
outside the boundaries of the Marine National Monument.
Yeah.
Okay.
Later in the cruise, we'll get a couple dives likely on some international water seamounts.
Ooh.
It's very exciting.
Yeah. Hopefully at least one. We might have had to eliminate one, but.
And then we'll be international sea
Oh,
explorers.
let's take a look at that coral. Yeah.
Video, let's push in.
Beautiful.
Yeah, it looks like a primnoid octocoral. So it has all those polyps are facing down, and when they're retracted, which they kind of are at the base, they have a really scaly appearance. Their sclerites are really large, so sclerites are like the small calcium carbonate components that make up some of the structure within octocorals, and the ones within this family are pretty pronounced. Looks like a very happy, healthy primnoid octocoral, though.
And we haven't talked a lot about our backgrounds, but Alexis, you study cold water coral, so this is like your baby right here.
Maybe not this one, but
It's coming full wide. What's
in
your
general.
favorite
Oh no.
coral?
I actually really love Paragorgia, the bubblegum coral.
Those are pretty.
That's pretty.
They're
that's
so
good choice.
cool.
Yeah.
They are really pretty.
We were on I was recently on the Okeanos last month or a month and a half ago, and our last dive of the cruise was in a canyon where at like the end of the dive we came to this like amazing bubblegum coral forest, essentially, where they were like meters.
Oh
they were
my
huge,
gosh.
and they were like pink ones and white ones and like lighter pink ones, and there was shrimp all over them. It was phenomenal.
It's like a 70s disco.
It was awesome. Yeah. But those ones are
Pushing back out.
super pretty.
I think it was off
down a bit
Johnston
for you.
Atoll. Speaking of like crazy environments, so off Johnston Atoll a couple of years ago, the
that
Okeanos
sure
found
is
like this
losing ground.
crazy big like essential forest of like these sponges and like these glass sponges hanging out that were super beautiful. And we're going to be going to Johnston Atoll later after these dives, so hopefully we'll see some really cool stuff out there too.
Yeah. Was that the Forest of the Weird?
It was the Forest of the Weird, and everyone should go Google Forest of the Weird.
If you haven't seen it already.
The weird. Do we need to like preface that with Johnston Atoll?
Maybe Johnston Atoll Forest of the Weird. I can't vouch for what pops up if you only Google Forest of the Weird, but it's really cool.
Yeah, there's a lot of really
I
interesting
have one. I
sponges
just
there.
never bring it upstairs.
It's very Seussian.
Yeah,
It's
Seussian.
Yeah.
Sorry.
It's very Seussian. It's very Lorax.
Classic Seussian.
Classic Seussian.
Renaissance Seussian.
Renaissance Seussian.
Wow, it is. Holy smokes.
Right?
So wait, with there's a sponge reef called Forest of the Weird. I'm sorry, I only caught half of that.
Yeah.
That is cool. That is the best name I've heard for a sponge reef.
It's a lot of stocked sponges, and the ones that look like they have like big eye. I mean, they look like big eyeballs. Sponges don't have eyeballs, but.
Cool. You probably have to wonder that Dr. Seuss would have a field day if he were alive and watching these feeds, you know?
Oh, man.
Especially like, this Iridogorgia, the spiral ones. It's just like, it doesn't get more Seussian.